I mean, I knew I was here 37 years ago, but to be honest, I never really thought about it until you just said that out loud. I'm not kidding, right? And I was like, it was a really sort of foundational place for me because you really got to commune with nature. We went diving all the time. My buddy at the time was Paul Nicklin, the National Geographic photographer, and so we took boats out and did 50 odd dives out there searching for cool animals, and always wanted to come back. And last year we were here for a stickleback meeting. We organized a meeting where people came from all over the world to talk about stickleback research. And we got a cool idea for this course, Rapid Evolution in the Anthropocene. That's what I'm teaching. And I'm here to tell you about a different angle for rapid evolution, okay? So the first thing to say is that we're going to do a thought experiment here. And there is Galapagos Eco-Evo stuff in this talk, which was the title. But I decided to give you a slightly different talk because this is the kind of one that makes me think the hardest. And so I'm hoping to make you think as well, in a good way. So the basic idea is that in order to understand the importance of evolution in structuring the world around us, that is rapid evolution that's occurring all the time, all around us, that the only way to really figure that out, to see how important it might be, is to do the thought experiment of asking, what if we turned it off? So with those words, I magically turned off evolution, okay? So we're going to see at the end of the talk what might conceptually have changed about the world around us, and then think about projecting that as we move forward. What would happen in 1 year? What would happen in 10 years, 100 years, 1,000 years, etc. So let's start from the last page of The Origin of Species, where Darwin sort of weaves this idea of complex ecological communities into evolution, and basically saying that those communities evolved together from a single ancestral origin. And it's fun, it's worth reading. It is interesting to contemplate a tangled bank. Here are some Google images of tangled bank, clothed with many plants of many kinds, with birds singing in the bushes, with various insects flitting about, and with worms crawling through the damp earth. And to reflect that these elaborately constructed forms, so different from each other, and dependent upon each other in so complex a manner, have all been produced by the laws acting around us. And he goes on to say that those main laws are growth and reproduction and natural selection. So what we're going to do is we're going to ask essentially what structures these communities, what maintains stabilities in these communities, why are they out there, and what will keep them there as we move into the future. Now, classically, when people consider communities, they just assume that this mouse is a mouse is a mouse, and that this rose-breasted grosbeak maybe is a rose-breasted grosbeak. No? It's a hawfinch. Is it a British thing, or it's not? Okay, it's a British thing. The hawfinch is not a rose-breasted grosbeak, right? So it's like a species level thing. You count up the species, you see how they're interacting, and you just assume that if it's a member of that species, that you know what it does, you know what it is, and you know how it will interact with everything around it. So it's a static thing, static entity created by past evolution. So evolution generated all that, but now it's this static place where interactions occur and evolution is essentially ignorable. And that sort of reflects this view that Darwin pronounced and that pretty much people accepted for hundreds of years later, or for 100 years long. We see nothing of these slow changes in progress until the hand of time has marked the long lapse of ages. The famous passage from The Origin of Species that people like to invoke when saying that Darwin felt evolution was slow, millions of years. Here's another example. She, meaning natural selection, can never take a leap, but must advance by the shortest and slowest steps. So there was this received wisdom from Darwin based on these words that evolution was something that happened a long time in the past, was critically important for generating everything around us, but basically it's so slow we can essentially ignore how it would affect the world around us on short time scales. And that really sort of persisted for more than 100 years. And then in the last 50 odd years, we've realized that there's a whole bunch of evolution that can occur in very short time frames. And these kind of contexts are the ones where it was first recognized and accepted. So the evolution of resistance to antiviral drugs, this is why it's hard to eliminate HIV. Antibiotic resistance, we all have heard about this. You know, make sure you finish your antibiotic treatment because otherwise it's going to evolve resistance. Chemotherapy resistance, that's why we die of cancer, because the cancer evolves away from our ability to control it. Pesticide resistance, herbicide resistance. So these things were all accepted probably by maybe the 50s, 60s, and the basic feeling was that, yeah, if you imply a really strong selective force, if you try to kill the thing intensely with some specific treatment, and it's a short-lived thing, so it can have a lot of generations, it's going to escape from your attempt to kill it, and evolution is the reason. So that's fine and everything, but the things that most people think about from a conservation biology, ecology perspective are the big things, right? We want to save, sea otters and whales and marbled murrelets and all these kind of things that sort of become these icons of conservation. And it turns out that in the last 20 years, we've realized that these sort of big, slow things evolve rapidly too. And that is sort of rejiggered the whole way people think about the problem, because it means that basically everything out there is evolving all the time around us. I happen to be a big fan of nature photography. These are some friends of mine over the years. I didn't have time to get a Bamfield one up. And then I also share a lot of camera trap videos on Instagram and places like that. So I really like nature, the big things, the cool, like exciting things, and they are evolving rapidly too. Okay, so now with that sort of introduction, let's go back to here. And let's ask, what is that rapid evolution going to do to these communities? How would we figure that out? Now, classic studies, what they do is they focus maybe on one of those organisms and they have some polymorphism in it, like it's different colors or different sizes or something like that. And then they ask, what's the effect of that difference in this one species? And it's big often. It has a huge effect on the things it eats, on the community of organisms that are parasites on it, the predators that interact with it. But that's just one thing. That's just one of these millions of organisms that are living in this community. So how would we ever understand how important rapid evolution was in structuring these systems? And I'd argue that the only way to conceptualize that is to do the thought experiment of asking what would happen if evolution stopped right now. And so that's the thought experiment that we're engaged in. And it's sort of riffing off this book called The World Without Us by Alan Weisman, where he did the thought experiment of turning off people, turning them off, just deleting people from the world, and then asking what would happen. So part of that's a thought experiment, but then you could also go to various places in the world, like Chernobyl, where basically people just sort of got out of there, and then ask how the physical structures decline, but also how the organisms change when people are gone. COVID-19 was another experiment in that, in some situations where people just didn't go certain places, and you saw a lot of changes in the organisms that were present in those locations. So I'm kind of asking the same thing. It's harder, but ask about the world without evolution. What would that look like? This is my daughter on a lava field in Hawaii. And to ask what would happen to the world if instead of removing people, we turned evolution off. So what I'm going to do is I'm going to go to 3 different places where the research that I've done has changed my thinking on this problem, and try to walk you through why I now think about the problem quite differently than I used to. And then by the end, we will have some idea of talking about how the world might change when we turned evolution off 5 minutes ago. So these are the 3 study systems I want to take you to. These are 3-spined stickleback, which a bunch of you know about, Trinidadian guppies, and Darwin's finches in the Galapagos. And this is all embedded in this conceptual way of thinking that I call eco-evolutionary dynamics. And that is the idea that ecosystems have communities of organisms that live in them, and those communities of organisms, each one of them is a population. And there's dynamics at the population level, the community level, and the ecosystem level that are all influencing each other. But every population in every community is composed of phenotypes. Those phenotypes are under selection and plastic effects, modifying the phenotype. some of which will evolve through genomic change. And then because these phenotypes are interacting with the environment, those evolutionary changes in the phenotype should feed back to modify the dynamics of that organism, the structure of the community in which it's embedded, and the ecosystem function that is embedded with the community. So I wrote a book about it. So I can hook you up later. I got some in the back of the trunk. So for real cheap. But actually, my favorite part of having the book was that this hashtag started called People Who Fell Asleep Reading My Book. So these are various evolutionary biologists and ecologists, Dan Simberloff, Dan Bolnick, Jonathan Losos, Mike Kinnison, Greg Bankman, and students and stuff who were having a good nap with my book. That was my favorite part. And so I have a copy here. So who suffers from insomnia? Sorry. So this book is actually for you. Because Haley won the competition for the most stickleback caught in a trap that we had yesterday in a very, very interesting way, which maybe she can tell you about later. So I will sign that for you later. Okay. Where's Braden? Sorry, man. Is Braden here? He was the leader for a while. Maybe that's why he's not here, because he knew he wasn't going to get anymore. Okay, so we're back here, right? So the first thing we're going to ask is, these organisms have phenotypes, there's populations of those organisms, and they're embedded in a community. So first, how will rapid evolution modify the phenotypes of these organisms? So to talk about that, we're going to go to the Galapagos and talk about Darwin's finches. Galapagos Islands are a small set of volcanic islands over a hotspot where lava erupted from the bottom of the ocean, basically built mountains under the ocean, and then they poke out the top. Never connected to the mainland. This is roughly 800 kilometers. So basically everything on the Galapagos got there by long distance dispersal from the mainland, or when humans bring them after that fact. So that means that it's a pretty depauperate community. There's not a lot of organisms there. When they get there, there's a lot of ecological opportunity, and you see dramatic, rapid changes that lead to the radiation of a bunch of organisms that do different things. So we studied the Darwin's finches, and just to give you a visual of what our research site looks like, so this is our tent where we basically are processing the finches. There'll be mist nets at various locations around here where we're catching the finches and then bringing them back to the research site where they get blood taken, beak size measurements, fecal samples, color measurements, bite force measurements, a whole bunch of different things depending on the goals that we have. Now, the primary thing that we study here is the interaction between finches and the seeds that they're eating. We're focusing on the ground finches. So here's a large beaked ground finch. Actually, it's a medium beaked ground finch, but it's a large version of it. He's picked up a maricarp, which is a woody fruit, and he's crunched it and opened up one little seed pocket and removed one seed. Now he's going to go for another one. Picks it up, moves his beak around a bit, scrunches it. He's got, oh, no, I didn't quite get it. Got to try the other side, work on it for a while. This is quarter speed, by the way. And okay, shoot, got it. Oh, damn it, dropped it. Okay, so the point of this video is that this is not an easy interaction for the plant or the seed. So if there's something that you can do as a plant, they're seed predators, they're not seed dispersers. There's something you do as a plant to stop that finch from getting into your maricarp to remove your seed, that'd be a good thing. But if you're a finch and you can come up with tools that better get you in there, then that should also be favored by natural selection. So you have this dynamic between the birds and what they're eating, both evolving in response to each other. So you have the result being a radiation of roughly 14 different species, all that are specialized on different food types that evolved within roughly 1 million years. Now, the cool thing is that in each of these systems, and how I want to talk to you about this idea of the tangled bank, is that you have these different effects and layers within the eco-evo framework that I gave you, and we're going to look at different components of that. So in each of these study systems, you can kind of populate this conceptual framework with real things that are happening in the community. So communities of seeds and plants, and the beaks of the finches and the genes involved in the beaks of those finches. So we spent quite a bit of time catching the finches. They're being held in those bags there while they're being processed. Sometimes film crews would come and lug their big IMAX 3D equipment out. This is my student, Luis de Leon, Fernando de Leon, who's now a prof at University of Massachusetts, Boston, who did a lot of this work. Now, if you know one thing about finches and rapid evolution, it's that they evolve rapidly, right? It's the classic example. Who's heard of like finches evolving rapidly? Probably more than half of the people in here. So their beaks change really rapidly in response to environmental change. And I know that because my mother bought me this book in Christmas 1995, which is the year it came out. And I read it, and I became an evolutionary biologist that day. Because I realized that you could study this process in nature, which in real time. And I had never thought of that as a possibility before. And here's just one example. This is work by Peter and Rosemary Grant, who started at McGill, by the way. They started this work at my university, McGill University. And they're showing changes in beak size through time in their study population. Here's one of the drought periods, and you see in this particular case, the beak size gets smaller. So it's in all the textbooks. It's our basic understanding. We know the genes involved in these rapid changes. And so that's like the big story. Environments change, finches change like that, right? What if we go just like 3 kilometers away to the population on the mainland and ask what they look like? They're completely different. That's the same species. So this change is big if you look at it in the perspective of that species. But if you go just 7 kilometers away, the finches are all, the same species are all bigger beaked. So what that really means is that they're evolving like crazy, but they're staying in the same place. So finches evolve rapidly, but they stay pretty much the same in any given place. So we did some thinking on this, and we basically wrote this paper, which we called the terroir of the finch. Anybody know what terroir is? It's the thing that make local vineyards good in winemaking. It's the power of place, essentially, in winemaking. My family is a winemaking family, so it just came to mind that basically the specific environmental effects that are present at any given place is what determines the size of the beak of the finch. Now, it might move around a bit, but it's not going to go very far from that local optimum. Now, you might think, okay, fine, but we need if there's some big environmental change that's consistent, maybe we should pick it up. Climate change, right? We all know that's having a huge effect. So let's see if the finches, over the last 20 years, we've been collecting data, have evolved in response to climate change. So there is a signature of increasing precipitation and of increasing temperature in Galapagos. So now let's track over that entire time frame, roughly 20 years, what's happening to the beaks of all the different finches in the different places, and see the rapid evolution in response to these climate warming. And there's nothing going on. The finches are staying the same for every trait, in every place, for every species. So no, they're not responding to climate change. Now, my argument then is the reason they aren't is because they're adapted to the seeds in the plants. And as long as the seeds in the plants are there, global change is not going to have a big effect on the evolution of the beaks. So we wanted to then ask, is this stability here the product of this idea that evolution is sort of keeping them in the right place? So it might go up in one year and down in another, but it's sort of orbiting around the right type for that local place. So we collected information on the beaks and survival of these 5 different groups. The small ground finch, the medium ground finch has 2 different beak morphs, the large ground finch and the cactus finch, which has an extended beak. So in a single community, in this adaptive radiation that's really the tips of the tree, they're all very closely related, all interbreed to some extent. Let's ask whether or not the changes through time in these populations are important and what is the reason for their stability or their change. So we collected a lot of data. So we have 17 years of data here, which amounts to 4,410 birds across the different beak-sized morphs and the different species. And what we asked then was, what does natural selection favor for a couple of the classic beak traits? The length of the beak and the depth of the beak. So using survival data, we built a landscape here called the adaptive landscape that has colored peaks that would come out that would correspond to fitness peaks that generated high fitness. So we're going to ask how many of those peaks are there and how close are the finches to those peaks. And as they change from year to year, how do they move in relation to those peaks? Okay, so there's going to be a landscape of color, and then the finch means for each of the species and morphs will be plotted on it. What do you think it looks like?
The 5 different morphs you have there are each going to correspond to a peak. 5
morphs, 5 peaks. That's what we would like. But why couldn't it just be flat? I mean, the funny thing is that nobody ever constructed these adaptive landscapes formally before because it's really hard to do. You need a ton of data, and you need to fill in the morphological space. So, are there 5 peaks? Are the finches close to them? You ready? You ready? Sure? Yeah, there's 5 peaks pretty much. Look at that. High fitness here, high fitness there, high fitness there, high fitness there, and a big ridge here. There's no peak. And the finches in any given instance are sort of moving around year to year in their average sizes, but they're never going far from the peak. So they're kind of just bouncing around the local peaks, and selection is basically keeping them in the long-term average place. So conditions change one year, their beaks get a little bit smaller, but then conditions change a bit the next year, their beaks go back. So what rapid evolution is doing is keeping them in the same place. That's the first key shift in my thinking. Rapid evolution isn't just about taking things to some new place. It's about keeping them where they are. So that's the point here. The main outcome of rapid evolution in generally and in finches in particular is that it's keeping them in the same place. So you're thinking like, wait a minute. In class, I saw some paper by Sarah Sanderson, your co-instructor, that said that traits are changing all the time, and you're going to see these meta-analyses with all these trait changes. So how does this idea that evolution is keeping things the same fit with this idea that there's all of these changes that are occurring? Well, it stems from that idea that basically evolution is just jumping all over the place. So basically, rapid evolution tends to take place on short timescales and show frequent reversals. So it's a very dynamic place. It's jumping all over the place, and it really doesn't accumulate to much unless there's some massive change in the environment. Okay, so back to the community, back to the Tangle Bank. So the first realization for me is that each of these different species here, what evolution is kind of doing for long periods of time is keeping them the same. It's buffering changes in the environment, interacting to changes around them, jumping around, but in general, keeping a mouse like a mouse and a chaffinch, hawfinch like a hawfinch, but not a rose-breasted grosbeak. Okay? Now, this community and the interactions, they won't just be determined by the traits of the organisms, they'll be determined by how many of them there are. So the next thing we're going to ask is how does evolution influence how many mice are there and how many chaffinches, hawfinches are there? Okay? So that's the next question. And to sort of explore how my thinking changed on that, we're going to go to Galapagos and talk about Trinidadian guppies. So here's Galapagos here in contrast to the, Trinidad here in contrast to Galapagos. It was previously connected to mainland Venezuela. So you have a relict fauna that is representative of the South American mainland. Now, if you go on the north slope of the Northern Range Mountains, you see this sort of we're looking at the top of the mountains, and then the computer's not fast enough to do it smoothly, but we're sort of going down from the hills down toward the lowlands close to the sea, and you're following the course of a stream there. And the forest here is quite nice, and then you find waterfalls. Right, see the waterfall here? So those waterfalls have a massive effect on structuring the communities that are present at these locations. Only some things get over these waterfalls, whereas other things don't. And in particular, guppies are pretty good. We have a lot of hypotheses for how this happens, but guppies are pretty good at getting over waterfalls. But their main predators are not. So there's a whole ton of work looking at how the guppies just above this waterfall evolve in response to having few predators versus the ones just below the waterfall evolving in response to lots of predators. And yeah, guppies pretty much look to some extent like simplified versions of the guppies in your fish tank. The females are swimming around, constantly trying to get food, and the males are swimming around, ignoring food, and constantly trying to mate with females. And so there's a whole bunch of work, this is in Trinidad, a whole bunch of work basically looking at the evolution of color, morphology, behavioral interactions in this natural system. So it's kind of like the finches. Basically, we run around. Some of the times we camp in the forest. Those are hammocks there for camping. And then we catch guppies, in this case using butterfly nets. And then we take them back to some processing station. In this case, a place called Simla, just on the south slope of the mountain range. We take pictures. We take clips for DNA. We take behavioral assays, look for parasites, etc. So, as I mentioned, the main contrast here was the one that everyone focuses on is high predation versus low predation. So, just as one example of evolutionary changes that occur in this system, including rapid evolutionary changes, there's color differences in the males depending on whether there's predators, nasty predators present or not. So, if there are nasty predators there, the males aren't that colorful. But if there are no nasty predators, the males get really colorful, because the females like to mate with colorful males. So John Endler in the 1970s did some experiments where he asked about how quickly this trait would evolve. And so he did a bunch of studies where he took high predation guppies and put them into new locations that did not have predators. And you saw the rapid evolution of increasing color in a whole host of these different parameters. So rapid evolution caused really dramatic changes when you change the environment by moving guppies from one place to another. And there's a whole cottage industry of people studying their favorite trait and whether it evolves and how rapidly in response to predation differences. So for example, think of your favorite trait. It differs between high and low predation guppies. Even if it hasn't been studied, it differs. Everything differs between these populations. But the question I want to ask is, how much does that evolution matter for their population dynamics? Not the traits anymore. We know that those change like crazy. But does that evolution matter for how many guppies there might be, for example? So we did sort of an additional experiment where we sort of leveraged an introduction that David Resnick had done, where there were guppies present in this river called the Yarra, but they were not present in this small river nearby called the Damier. And there was a waterfall here. They weren't present above or below the waterfall. So David introduced them from that ancestral population into the populations above and below the waterfall. Okay, so you have them coming from here, being put into these two locations, and then left there to evolve. So we introduced fish from the one site into two others, and then he waited 7 years, and then we said, well, okay, you haven't studied them yet. We would like to measure not how just the traits are changing, but how their survival ability is changing. We're trying to get closer to how the evolution will influence the dynamics of these populations, their persistence, their growth, their spread. So we then said, okay, well, what we'll do is we're going to test how much these guys have evolved to do better in these local environments in now 7 years relative to their ancestors. So we did a new experiment where we took guppies from each of these 2 sites and we marked them, and then we released them back into those sites alongside individuals from their ancestral population. So now we're competing in each of these 2 derived sites, the populations that have had the privilege of evolving there for 7 years, roughly 25 generations, to their ancestral populations who have not been evolving there. You see the experiment? We want to know how much better these guys do after the 7 years than their ancestral genotypes. So the data is going to look something like this, right? So you've got site 1 up here, and there's going to be some survival estimate for the derived population that's been evolving there for 7 years, and you'll have some survival estimate for their ancestral population. And then you'll have the similar thing for the other site. So which bar would you expect to be higher? Which bars? Which one, sorry? The ancestral, so you're postulating that the ancestral population will do better than the ones that, right, so the red bar will be lower, right? The green and blue should be higher. That make sense? So the local fish adapting there for 7 years should now do better there than the genotypes that were put in there 7 years ago that had not evolved there. But how much higher? I don't know. Like, no one had done an experiment like this, so we had no idea. So, I mean, what do you guys think? Like, is it going to be, I don't know, 10% higher, 50% higher? You know, I don't know. See, so who thinks it's going to be like, we'll do 10, I don't know, 30, 50, and 70, okay? Who thinks it's going to be 10% better after 7 years? Okay, good, because usually people don't pick anything on either end. 10, what did I say, 30% higher. Okay, that's a lot, right? 30% in survival prospects in 7 years, 50% better. 1 person, 2, and 70% better. Oh, usually people don't pick the ends. Okay, you ready? 50% higher. Okay, so the traits change pretty dramatically, but there's roughly a 50% improvement in their survival prospects in those environments. So the point of this is that what rapid evolution does is it doesn't just change the traits, but those trait changes combined with everything else you can't measure change how well they do their fitness locally. So what does this mean then for thinking back to, there's a whole bunch more work on this, blah, 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 we publish papers, etc. So what does that mean for this then? It means that evolution, that jumping back and forth evolution, it's not just keeping the traits the same, but it's likely keeping the numbers of individuals the same. So this is this sort of interaction, this mutualism, competition, predation, they're all responding to each other. Their traits are sort of like staying in that, or somewhere around, orbiting around that best trait value to be. They're interacting with each other, and evolution is stabilizing the whole effing thing. So evolution is keeping traits the same, and it's basically keeping all those population abundances there. So if you turned off evolution, probably these things are going to start going extinct, right? Because they can't respond. Okay, so finally, the final vignette, I guess, is stickleback. And for stickleback, I want to now go to this final thing. So we've asked about how phenotypes change and how rapid evolution influences that. And we asked about how rapid evolution influences population dynamics. But phenotypes in say, predators can influence their consumption of prey, for example. So now we're going to ask about a direct interaction here, where changes in the phenotypes might influence the community of organisms that they interact with. So it's not just, we're moving beyond the focal organism now. We're asking how changes in a focal organism will influence everything else. So we need a trait here that's going to be good for linking changes or trait stability to community structure. And a good thing is the business end of the stickleback and how they consume prey. So we're taking a 3D CT tour backwards through the mouth of a stickleback, some gill rakers here, which you guys will be measuring or looking at, coming out through the eye and basically looking back. So this is the business end of the stickleback, and it influences what they feed on. So if that evolves, it should change what they feed on, which would change the community of things they feed on. So that's the premise here. So we want to focus on foraging traits, look at the diversity of stickleback, all which evolved post-glacially from marine ancestors. Stickleback are incredibly diverse in all these different populations in freshwater as a result of adaptation to those environments. And we're going to look at how these foraging traits and their changes, their rapid evolution, will influence zooplankton and benthic macroinvertebrate communities, the things they feed on. So we're especially leveraging this contrast between feeding on the bottom of lakes, usually still shallow, which is called benthic, and they feed on benthic macroinvertebrates, so insect larvae mainly, on the bottom of the lakes, usually again in shallow water, versus the ones that are out in the open water of the lake feeding on zooplankton, small crustaceans swimming around in the middle of the lake. Now, there's a bunch of stickleback foraging traits that evolve in response to this limnetic versus benthic contrast. And the one I'm just going to specifically focus on here is the gill rakers. So those were the projections you saw inside the gill arches. They sort of stick into the, along the buccal cavity, and they influence sort of what happens to prey that comes in the mouth. So water's coming in the mouth, prey's coming in the mouth. You want the water to go out the gills, but you don't want the food to go out the gills. So it kind of acts like a sieve almost. So if you eat small stuff, you want a lot of gill rakers, and you want them to be closely spaced. If you eat big stuff, they're going to get in there and destroy all those gill rakers. So you want fewer that are more widely spaced. And it's very strongly genetically determined. Okay, so I'm just going to tell you 3 different things about this. The first point is that these traits, these trophic differences, evolve in response to foraging opportunity differences. So that it's a very predictable effect. We know that this benthic limnetic contrast works well. And to show you that, we're going to go to Bamfield. So here's Pechina Lake, Frederick Lake, and then a bunch of other lakes on Vancouver Island. And we're basically going to look at the stream fish. There's almost no zooplankton in streams, so we expect them to be exclusively or almost exclusively benthic foragers. But in lakes, they could vary from feeding on all zooplankton to feeding on mostly benthos. But they're going to be way more limnetic feeding in lakes than in streams. So 16 different lake stream pairs, and we can look at, for each system, every one of those is independently colonized from marine ancestors. So it's 16 evolutionary replicates of lake stream divergence. We're going to ask how many gill rakers do they have in the lake. We would expect them to have more gill rakers versus the stream, where they should have fewer gill rakers because they're feeding on bigger stuff, benthic stuff. And I don't have to do that, because I probably wouldn't show you the data if it didn't show that, right? So each of these lines is a different watershed, and you see that within each one, the stream fish have fewer gill rakers than the lake fish. So there's a very predictable trait change in response to environmental shifts. But does it evolve rapidly? So here, we're just going to jump up to Haida Gwaii, just north. There's Vancouver Island here, obviously. There's Haida Gwaii up there. Lots and lots of stickleback on Haida Gwaii. Tom Reimken, who will give a seminar in exactly 1 week from now, will talk about his work, probably, I assume, up on Haida Gwaii. Don't miss it. And one of the big lakes there is called Mayor Lake. And what Tom did, hell, maybe he'll even talk about this. But we have new data, so it won't be the same data. This big lake called Mayor Lake, and he took stickleback from this hyperlimnetic lake where they're really specialized on feeding on zooplankton and avoiding big predators and all of that. And he took them and put them in a new environment where you might expect gill rakers to evolve rapidly. He put them in a benthic environment. Now, how striking is this contrast? We'll just follow as the Tom walks, taking his bucket of stickleback, moving out. That's the whole lake up there. So it's a huge, it's a big lake. And he took those poor limnetic specialist stickleback and put them in that. So that's probably shallower than Hummingbird Pond where we're going to be working. So
Stickleback in there before?
there were not. It's an old, it was a quarry where they quarried gravel to build the road. So there were no stickleback in there. And Lucas and Sarah have done genetic work to show that these fish did indeed come from Mare Lake. But Tom didn't do this just once. He did it a bunch of times. So he took the Mare Lake fish and put them in the pond. He also put them in another pond shown right here. There's another little tiny pond. But he also took him from the lake where he lived, Drizzle Lake, and put him in a little tiny pond. So this is the distribution of gill rakers in those two lakes. And the question is, do they evolve to be fewer when you put them in the ponds? Yes. Okay. But it's not instantaneous. It takes a while. So this trait has a strong ecological effect. It's very predictable, and it can evolve rapidly. But will the variation in this trait and its evolution change entire prey communities in lakes? So the final thing I want to tell you really quick is now continuing further north. This is the Kenai Peninsula in Alaska. And we're going to ask, do these benthic limnetic stickleback ecotypes really matter for entire lake prey communities? So we're going to take fish from particular lakes that are either benthic or limnetic populations, and we're going to put them in new lakes and see if they change the zooplankton in those lakes. But more importantly, do the benthic type stickleback change those prey communities in lakes in a different way than the limnetic ones? As a quick sidebar, we were able to do this because this is the actual landscapes. These are real lakes. These are not those cute little experimental ponds that people are so excited about. These are real lakes on the real landscape. And the reason why we got to do this experiment was because there was an invasive species in these lakes, the northern pike. And it eliminated all the stickleback in those lakes. They wanted to get rid of the pike because they didn't want it spreading to places where it would start eating salmonid fishes. And they wanted to have the Kenai Peninsula free of pike. So they put rotenone, chemical poison, plant-based chemical poison, into all of these things to kill everything in them. And they're empty. Now, they're not really connected with major streams. So if you want to have anything happen with respect to getting stickleback, the native species back in here, you got to put them back in yourself. And normally the Alaska Department of Fish and Game just dumps any old thing in there. But we heard that this was happening, and we said, well, can we do it? And they said, well, if you want to do the work, sure, go ahead. So in a very short period of time, we got together a team to basically do this experiment. And there's a whole bunch of stuff involved here, but for the purposes of this talk, we put benthic fish in these three places, and we put limnetic origin stickleback in those these four places. We also mix them in other lakes, and there's a whole bunch of stuff about that. But these lakes got limnetic fish, lakes/ponds. These ones got benthic fish. Does that have a repeatable signal of those ecotype differences in structuring these communities in the real landscape? So the data look like this. This is an ordination of diet space. So these are principal component combinations of different prey types in the zooplankton. And you've got your different lakes here. These are the ones that got benthic fish. These are the ones that got limnetic stickleback. And these are the lakes that got both types of stickleback, which I didn't show. So there's going to be a bunch of points on there that correspond to different samples from the lakes. And the question is, in that zooplankton community space, do the benthic and limnetic lakes differ? Now, I can't emphasize how we had no clue. I think there's a ton of reasons why there should be no effect here. You know how you're supposed to standardize in experiments? You're supposed to control for variation, right? That's what they tell you. If you want to know if benthic and limnetic fish have an effect, you got to control for everything else. We controlled for nothing. So we are really asking if there's a big enough signal to emerge beyond all of that noise that people usually get rid of. And I had no idea if there would be. My God, look at that. There's a big effect. So each of these is 4 points sampled from the lakes. We have data from multiple years. And the lakes that got benthic fish are here. They have a different prey community than the lakes that got limnetic fish here. And yeah, the lakes that got both types are intermediate. Like, it's just ridiculously good. So it's just incredible to think that these two different stickleback ecotypes, which evolve predictably, can evolve rapidly, will also, as they evolve, cause dramatic differences in prey community structure within lakes. So in this experiment, we have the opportunity to keep following it moving forward and basically study how these interactions occur. So now, as they evolve in those lakes, Claire is doing some work on that, showing that they're evolving within these lakes. And Hannah is also involved in that work. And Braden, I don't know where he is, but he's also involved in this work as well. Well, okay, Lucas is involved in this work, and Sarah's involved in this work, and pretty much everyone's involved in this work. So these are a couple of lakes. It's a good thing I said Sarah, because that's Sarah right there, right? Yeah. So these are two of the real lakes where we put the stickleback in. So obviously, this kind of experiment, there's a lot of stuff we want to measure, and so there's a ton of people involved. These are just the PIs, Stan Bolnick, who does a lot of work out here, Katie Peichel, Rowan Barrett, Rini, your niece, your aunt. That's her right there. And I didn't add her because I knew you'd be here. She was on there originally. Lots of people involved in this experiment. But you know, when I put this up and I was kind of like going through it in the lab, I realized that the people who actually do the real work, who are like the pointy end of the spear, are who? The graduate students, right? Which are basically Lucas. You know those? Have you been seeing those memes recently of Erling Haaland, and like it kind of looks like, doesn't it look a little bit like? And Claire, who are your TAs, who is kind of pulling the same pose completely coincidentally. Okay, so my contention is that the main thing that rapid evolution is going to do is it's going to keep traits roughly the same, it's going to keep population dynamics roughly the same, it's going to take communities roughly the same if that evolution keeps things where they are. Okay? So now, conceptually then, what happened, do you think, when we turned off evolution an hour ago? 55 minutes ago. What was the first thing you would have noticed?
Probably nothing at this point.
Okay, I will start then. I think that this room will smell different because the bacteria in this room, their dynamics are going to be completely messed up by the fact that they can't evolve in response to them and the changes in heat and the changes in temperature. If bacteria have resources, they're going to divide almost continually. So we might even see little piles of bacteria growing in the corners of the room where we left an orange or something that they can feed on, right? What about tomorrow morning? What do you think you'd notice? I think I'd have a stomach ache, don't you? Because my gut microbiome community is going to be quite different. You know, the stickleback in those tanks down there might die because the microbes in them have modified them. Okay, what are some other things that might happen in the next year? Nobody? Sorry, wait. Extinction of what? What's going to go extinct?
Something that probably has a shorter lifespan.
Yeah. A lot of zooplankton have many generations within lakes, and it turns out that they evolve incredibly within a single year in response to changes in predation, changes in temperature. There's a huge bunch of clonal generations that zooplankton go through that are adaptive. There's many insects that have multiple generations per year for which the same thing will happen. You see huge allele frequency shifts across many places in the genome over just a span of months. That's all done with, right? So, you know, the bacteria, the mosquitoes, maybe, no, the mosquitoes only have like one generation per year, maybe. I'm not sure actually. But a lot of like the fruit flies, they're going to be different, right? There might be more of them, there might be less of them. They might eat your fruit less or more. What about in, I don't know, let's say 10 years, or let's say I have a few years, 5 years, 10 years.
extinction cascade and there's just nothing, period?
Well, okay. At some point, yes. That's where we're headed, right? First, I'd say in like 100 years, you're going to have a bunch of old growth trees around and nothing else, right? Because there's basically no ability for any organism to respond to any environmental change anymore in an evolutionary sense, or to any other changes in the community. Parasites are going to cause hosts to go extinct, and then they're going to go extinct. The parasites on the parasites will then go extinct. So yeah, I think you'd get this extinction cascade, and I think we would see differences the end of this summer, and I think it would look a dramatically different world by next year, and we would not recognize the place in 10 years. So that's my hypothesis, and I think that the only way to sort of think about this role of rapid evolution is to try that kind of experiment where you say, what would happen if it just stopped working? Because there's no way we can do an experiment that will replicate all of those evolutionary contributions to maintaining the world around us. What rapid evolution does is it keeps things working, keeps them the same, roughly, and it keeps things humming along. If you turned it off, it'd be over. So that means that rapid evolution is an incredibly important force structuring the world around us and keeping us happy and healthy or unhealthy. Evolution is just doing a ton of stuff right now all around us. And this is my own personal way of thinking about the problem, because I had just sort of thought about the change part without realizing that the primary role of rapid evolution through these changes was to keep everything the same. Thank you. A couple minutes for questions, I guess. I tried to make it shorter, but I just